Let's take up some important points about biomolecules. Key points: monosaccharides and disaccharides represent only small fraction of total amount of carbohydrates in natural world. Polysaccharides are found to more extent. In recent years, sucrose has been replaced in many commercial products by corn syrup, which is obtained when corn starch is broken down. Corn syrup is primarily glucose, which is only 70% as sweet as sucrose. Almost without exception, the monosaccharides are optically active. Most of them found in nature have D configuration. The simplest aldose is glyceraldehyde, which is also optically active. Only dihydroxyacetone, which is isomer of glyceraldehyde, is not optically active. Fructose is about 2.5 times as sweet as glucose. A commercial process is therefore developed which uses an isomerase enzymes to convert about half of the glucose in the corn syrups into fructose. The high fructose corn sweetener is just as sweet as sucrose and found extensive use in soft drinks. Fructose is the sweetest sugar, lactose is least sweet sugar. Fructose contains keto group, but still it reduces swelling solution because under basic condition fructose undergoes rearrangement and forms glucose and mannose, which are reducing sugar. This rearrangement is called Lobry-de-Bruyn when Inkenstein rearrangement. Molish test is used for detection of all kinds of carbohydrates. Molish reagent, 1% alcoholic solution of alpha-naphthol is added to aqueous solution of carbohydrates, followed by concentrated sulfuric acid slowly along the sides of the test tube. A violet ring is formed at the junction of two layers, which confirms the presence of carbohydrate. Points to remember: the pH at which there is no migration of amino acid under the influence of electric field is called isoelectric point. Why there is no migration? Because positive charge and negative charge on the amino acid is equal, so it is neutralized. Out of 20 amino acids, 10 required for protein synthesis are called non-essential or dispensable amino acids because these are already synthesized in our body. Whereas remaining 10 amino acids like valine, leucine, isoleucine, phenylalanine, methionine, tryptophan, threonine, lysine, arginine, and histidine, which cannot be synthesized in our body, are called essential or indispensable amino acids. They must be part of our diet. Their deficiency may lead to the various diseases, and it also will suppress the growth and development of the body. The lack of amino acid in the diet, protein deficiency, causes disease known as kwashiorkor. The amino acid unit having free amino is called N-terminal, whereas carboxylic acid group is called C-terminal. Chemically, proteins are complex nitrogenous compounds. For growth and maintenance of life, they are condensation polymer of amino acids, and proteins on hydrolysis will produce amino acids. Congenital disease, phenylketonuria, is caused by deficiency of enzyme phenylalanine hydroxylase, while albinism is caused by deficiency of enzyme tyrosinase. Streptokinase is used to dissolve blood clots in case of heart attacks. Raffinose is a trisaccharide and on hydrolysis it gives three monosaccharides: glucose, fructose, and galactose. Stachycose is a tetrasaccharide. On hydrolysis it gives 1 mole of glucose, 1 mole of fructose, and 2 moles of galactose. Hydrolysis is carried out in presence of dilute acid or HI. Alpha glucose has specific rotation of 112 degree, beta glucose has 19 degree, so they differ in optical rotation. When either of the two is dissolved in water and allowed to stand, they get converted into equilibrium mixture of both with small amount of open chain form. So equilibrium mixture contains around 36% alpha glucose and 64% beta glucose. Very small amount, 0.2% is open chain. As a result of this equilibrium, the specific rotation of alpha D glucose decreases from 112 to 52.7, whereas beta glucose increases from 19 to 52.7. This change in optical rotation to a specific value at equilibrium is called mutarotation. A plastic celluloid is obtained when pyroxilin, a mixture of cellulose dinitrate and small amount of cellulose mononitrate, is mixed with plasticizers like camphor and alcohol. Colloid oil is solution of cellulose nitrate or pyroxilin in ether and alcohol and is used for manufacture of lacquer. It is also used to decrease the pore size of filter paper and convert ordinary filter paper into ultra filters. Rayon is the chemical name given to all fibers made from chemical treatment of cellulose. Acetate rayon is cellulose acetate. It is also known as artificial silk or fiber silk. Viscous rayon is cellulose xanthate. Proteins can be qualitatively tested by Biuret test, Millen's test, xanthoprotic test, and Ninhydrin test, which we are going to discuss. Biuret test: on adding a dilute solution of copper sulphate to alkaline solution of protein, a violet color is developed. This is the test. This test is due to the presence of peptide linkage. It can be used for all proteins. Xanthoprotic test: some proteins give yellow color with concentrated nitric acid. You must have seen that if nitric acid comes in contact with your skin, it becomes yellow. The upper part becomes yellow. After some time, this skin will go away and new skin will come. The formation of yellow color is due to the reaction of nitric acid with benzenoid structure, which turns orange. This color becomes orange on addition of ammonium hydroxide solution. Millen's test: when Millen's reagent, mercurous chloride and mercuric nitrate in nitric acid, is added to protein solution, a white precipitate which turns brick red on heating is formed. This test is given by proteins which give tyrosine on hydrolysis. This is due to presence of phenolic group. So this test has limited use. Only specific proteins respond to this test. Ninhydrin test is given by all proteins. When protein is boiled with dilute solution of ninhydrin, a violet color is obtained. So this can be used for titration to find out the content of protein in various fruits, vegetables, and other soya bean, etc. The monosaccharides which differ in configuration at C1 in aldose and C2 in ketose are called anomers. For example, alpha-D-glucose and beta-D-glucose are anomers. Similarly, alpha-D-fructose and beta-D-fructose are anomers. Anomeric carbon atom is also known as glycosidic carbon. The monosaccharides which differ in configuration at the carbon other than the anomeric carbon are called apimers. For example, glucose and mannose which differ in configuration at C2 are called C2 apimers, whereas glucose and galactose differ in configuration at C4 carbon, so they are called C4 apimers. A nucleotide consists of three components: base, sugar, and phosphate, and has structure base-sugar-phosphate. A nucleoside has only two components: base and sugar.